So we've all seen microscopes before, and hopefully you've used them in your science lessons, and essentially the way they work is by utilizing magnifying lenses. And when you look through the microscope, essentially the cells or whatever you're looking at tend to be blown up and look much bigger, and you can look at them in a lot more detail. So what's actually going on? Well, generally we have two types of microscopes, and the first microscopes that were developed were called light microscopes, and these are the ones that you'll generally see in your classroom. And the idea is that light rays will come through here, will bounce off of our cells and go through the magnifying glass to be seen in human eye, your eye over here, and these light rays will then travel the eye. And actually this allows us to then see the cells, and because of the magnifying lens, it magnifies the cells, the images that we see, and they look a lot bigger, and we can see our cells or things like that. Things are very, very small, and be able to draw them and understand how the subcellular structures look like. So this is actually allows us to see subcellular structures. But over time, as technology evolves, we developed more sophisticated microscopes. So we use things now in very sophisticated labs called electron microscope, and the idea is that light has a certain wavelength and electrons have a much smaller wavelength than light. So because they have a smaller wavelength, what this means is that it increases the resolving power and increases the resolution. And essentially what that means is that we can see at an even smaller scale, and we can differentiate between even smaller objects. And so the way an electron microscope works, very similar to a light microscope, but instead of having light, you basically replace the light with electrons. Okay, so this allows us to see basically in finer detail and understand more subcellular structures. Okay, and so if we have a microscope, then we see our images at a different size than our object. So we do need to know how to actually convert between the size of the image and the size of the object. And so we can use a simple formula, which hopefully you've seen before, like the magnification is equal to the image size over the object size. Okay, and we should be able to comfortable using this along with standard form. And so let's do some questions to actually test ourselves out on it. Okay, feel free to pause the video and have a go as usual. So question 1, how big will a 20 centimeter object look through a 500 times microscope? Let's actually write our equation out. So the magnification is equal to the image size over the object size. So I'm just going to shorten it, but you should know the full thing. And so we can write that we have a magnification of 500 times. That's going to be equal to the image, or I, divided by the object, which was 20 centimeters. And so the image size is going to be equal to 500 multiplied by 20, and that will give us 10,000. So our image will look absolutely huge. It will look 10,000 centimeters. Question number 2. Why do electron microscopes have greater resolving power than light microscopes? So we'll just write it over here. Essentially, electron microscopes are using electrons, and electrons have a lower wavelength than light. So because they have a shorter wavelength, that gives them a greater resolving power than light microscopes, so we can see things in finer detail. Okay, so hopefully that all makes sense, and I will see you guys in the next one.